Assalamu alaikum and welcome to our new video about Streptococcus pneumoniae. So firstly, let's look at how we diagnose a Streptococcus pneumoniae infection in the lab, what kind of tests we need to do, and then we'll look at the diseases that it causes. So in the lab, the first thing you can do, this is your tissue sample, your sputum or blood or whatever it is, and you can do a gram stain on it. And what will the gram stain show? So you can see with a Streptococcus infection, and this is Streptococcus pneumoniae, you will have gram-positive cocci, and strep pneumoniae are arranged as diplococci. So what you'll see on the gram stain are gram-positive diplococci. And remember with the strep, if you do a catalase test, it'll be catalase negative. So there'll be no bubbles on the test. And we explained that in our previous video. So if you see this picture under the microscope, you'll know this is a streptococcal infection. But there are at least 6 different types of streptococcus that cause disease. So how are we going to differentiate between them? Well, now that we know it's streptococcus, we can culture it on blood agar, and the streptococci are divided into groups based on how they break down RBCs. So 2 of them are alpha hemolytic, which means they partially break down RBCs. 2 of them are beta hemolytic, so they cause complete breakdown of RBCs. And 2 are gamma hemolytic, so they don't break down RBCs. So there are 2 streptococci in each category. This is the first step in classifying streptococci. And strep pneumonia is alpha hemolytic, so it causes partial hemolysis. Now let's look at how it looks on a blood agar plate. So this is our blood agar, and there are 2 kinds of strep that are alpha hemolytic, and there are 2 kinds of strep that are alpha hemolytic. So there's strep pneumonia and strep viridans, and both of them will cause partial hemolysis. And apparently you're supposed to see a green area or green halo around the colonies, where that black arrow is, for example. Even if it doesn't look green, just remember that partial hemolysis will give you green colonies. So now we know it's alpha hemolytic. It's either strep pneumonia or strep viridans. And there are 2 tests we can do to differentiate between them. The first one is called the optochin test. Now optochin is a chemical, it's kind of like an antibiotic. And strep pneumonia is sensitive to this chemical. It kills strep pneumonia. So if you grow colonies, as you can see on the right are colonies of strep pneumonia, and they can't grow near the disc that has optochin in it. So strep pneumonia is optochin sensitive. On the other hand, on the left, you can see that strep viridans grows right around the disc of optochin, so it's optochin resistant. Now the second test to differentiate between these 2 is the bile test. So strep pneumonia is sensitive to bile. Basically bile dissolves it, whereas strep viridans is resistant. The way you do this test is you mix bacteria into a suspension, into a fluid, in a test tube, as you can see, and the tube becomes cloudy, like the tube on the left, and then you add bile to it. If the bile dissolves the bacteria, you will get a clear fluid, like the one on the right, and this tells you that the bacteria in there is sensitive to bile. It's being dissolved by bile, so that is strep pneumonia. If it remains cloudy, then it's strep viridans. Okay, now based on these 2 tests, we know that we have strep pneumonia. There's a third test we can do on strep pneumonia, and it's based on the fact that strep pneumonia has a capsule, and this is very important to remember. And it's called the Quelling test, or the Quelling reaction. So basically in the Quelling reaction, shows that, so basically the Quelling reaction shows one, that strep pneumonia has a capsule, and secondly, it's actually used for serotyping. So what happens in this test is you'll mix the strep pneumonia with antibodies against this capsule, and when you look at it under the microscope, the bacteria are all swollen up like those four on the bottom, and that tells you that firstly that this has a capsule because it was swollen, so that's strep pneumonia, and secondly it tells you the subspecies of strep pneumonia. The way I remember it is Quelling swells up with water, so strep pneumonia is Quelling positive. Okay, let's recap what the different tests that we can do for strep pneumonia. Firstly, it's gram-positive diplococci. This is on the gram stain, and it's catalase negative. All the strep are like this, by the way. They're gram-positive diplococci or streptococci, and they're catalase negative. Specifically for strep pneumo, it's alpha hemolytic, so partial breakdown of RBC. It's opticin sensitive, and it's bile soluble. And the last one is strep pneumonia is Quelling positive. Okay, now let's look at the diseases that strep pneumonia can cause. So it can cause four main diseases. And first one is meningitis. It's the #1 cause of bacterial meningitis in adults. It can cause pneumonia from its name, pneumoniae. It's the #1 cause of bacterial pneumonia in adults. It can cause otitis media, which is infection of the middle ear, and it's the #1 cause of this in children. So you can see it's pretty serious about its infections. It likes to be #1. And it causes sinusitis. So there's a cool mnemonic. It's found in first aid, so thanks to them. We can remember these four diseases. You can put it in a mnemonic called MOPS. M is for meningitis, O for otitis media, P for pneumonia, and S for sinusitis. And the cool thing about MOPS is it also helps you remember that strep pneumonia is most opticin sensitive. And yeah, the way I remember MOPS with strep pneumonia is, strep pneumonia has a gangster name, and his name is Streptococcus Pneumoniae. And it has two virulence factors. The first one we already talked about is the capsule. Without the capsule, it can't cause disease, and capsule gives it Quelling positive reaction. And secondly, strep pneumonia has IgA protease. Now, IgA protease, that means it has a protein or protease that breaks down IgA. Here's our famous cell membrane, and this is the IgA protease, and it breaks down IgA antibodies. So clinically, remembering this is actually really useful, because remember, IgA is the main antibody for mucosal immunity. So if the bacteria has an IgA protease, it means it will infect mucosal surfaces. And if you look at the MOPS, the diseases caused by strep pneumo, it causes sinusitis, pneumonia, and otitis. All these have mucosal surfaces. So this is where the bacteria naturally causes disease. So that's going to help you remember that it has IgA protease. Thank you so much, and see you in our next video about strep viridans.